Assalamualaikum warahmatullahi wabarakatuh. Today, I would like to present my research about assessment of phosphate solubilization and indole acetic acid production of phosphate solubilizing bacteria isolated from acid soils Lampung, Indonesia. I am Muhammad Asril from Department of Biology, Institute Teknologi Sumatera. My research background is phosphorus is a macro-essential nutrient needed by plants such as carbon and nitrogen. Plant obtain phosphor element entirely from the soil or root exudates. The amount of phosphate in the soil is strongly influenced by soil condition. On marginal dryland, especially old soil, which are often found in Indonesia, namely high soil acidity level, such as in Lampung or Institute Teknologi Sumatera's area. This situation cause low available phosphor and high aluminum, calcium, and ferrum content. So the low availability of phosphor is due to high phosphor fixation by these minerals. So difficult to be absorbed by plants. Plants use phosphate for only 10 until 30% of the phosphate fertilizer that is given. So one way to increase the efficiency of phosphate fertilization is to use phosphate solubilizing bacteria. Phosphate solubilizing bacteria can dissolve phosphate that is difficult to dissolve from soil or fertilizer to dissolve and be available in the soil so that it can be absorbed by plants. In addition to the ability to dissolve phosphate, the production of hormone, which is indole acetic acid by bacteria, is an important criteria as a plant growth promoter. Hasil analysis 2020 isolated various isolate from acid soil from Institute Teknologi Sumatera areas, Lampung. Some of these isolate have not been characterized in more detail regarding the potential for phosphate dissolution and EA production, which can be used as a potential isolate to increase plant growth. So the study aimed to detect or assess qualitative and quantitative potential phosphate dissolving tree isolate like AF and AP5, 7, and 10, and its ability to produce indole acetic acid. This is method using in this research. And the result is, the first one is characterization of phosphate solubilizing isolate. We can see that the tree of isolate had less variable characteristic. Based on biochemical test, the tree isolate also had not varied characters. So, but indifference is gram AFNAP5 and AFN7 is gram positive bacteria in the form of coccus. AFNAP10 is gram aggregative in the form of coccus. So, and after that, point of my research, qualitative estimation of phosphate solubilization, you can see based on phosphate dissolution test on solid Picopskaya medium, the tree of bacterial isolate were able to dissolve calcium phosphate indicated by the forming of clear zone around the colony. You can see clear zone, clear zone. After 7 days of incubation, various clear zones were formed. The FNAP7 isolate the highest solubilizing index of phosphate compared to other isolate. The formation of clear zone indicated the activity of the colony produce organic acid, which are able to bind with the calcium ions to form calcium phosphate compound in Picopskaya medium and free hydrogen phosphate ions to form clear colors area. Various bacteria has been reported to have various solubilizing in that phosphate like Pseudomonas or Bacillus. But the ability of bacterial phosphate dissolving can be seen from the phosphate dissolution index. The clear zone on solid medium cannot show the ability of each bacteria to dissolve the amount of dissolved phosphate. This is because the clear zone form is an early sign of the presence of phosphate dissolution by bacteria. Salvi et al. 2017 also reported that the ability of bacteria to dissolve phosphate qualitatively in solid Picopskaya medium will be different in quantitatively in liquid Pickup-Skaya medium. So after that, we confirm in quantitative estimation of phosphate solubilization index. We can see the three isolate had high activity EF and AP7, highest phosphate dissolving activity of 392 compared to EF and AP10 and EF and AP5. This confirmed that high phosphate dissolution in solid medium is not necessarily positively correlated with high phosphate dissolution quantitatively. Changing the sulfate phosphate concentration in liquid culture can occur due to deposition of phosphate from organic metabolite or the formation of organophosphate compound with organic acid secretion. The quantitative test we can see of phosphate dissolution from isolate showed that the maximum phosphate dissolution observed on this third incubation in the FNAP10 and the AFNAP7 isolate had the highest activity on 4th day, and AFNAP5 isolate had the highest activity in 7 days of incubation. So beside of the organic acid secreted by bacteria contribute to dissolution of phosphor, 3 factors play a role in this process, namely pH, type of organic acid, and environmental acidity feedback. Bacteria will release organic acid during the phosphate dissolving process, thus lowering the pH of the medium, which indicated the dissolution of phosphate by bacteria. So that dissolve process runs well. During the phosphate dissolving process, it was seen the pH of medium changed with different patterns. The FNAP5 and the FNAP7 had the same pH change, with increase with the length of the incubation period, while the EFNAP10 also decreased in pH along with the length of incubation period, and increased again after 6 days of incubation. Naturally, during the phosphate dissolving process, the bacteria will produce organic acid, thereby lowering the pH in the medium, which indicate that the dissolving process is complete, as shown is ISO-DF and NIP10. However, change in the pH of the medium are, as reported, not applicable to ISO-DF and NIP5 and AFN7 and AP7. Like Mara et al. 2015 reported that there was no correlation between the decrease in pH and the value of phosphate dissolving by bacteria. And after that, during process solubilizing in quantitatively, you can see the pH of an AP7 and AP7 isolate and 10 was in the pH range acid category. This is not a pH not too different from the soil sample used as a source of isolate, which is 4.09. Apart from being a phosphate solubilizing, another potential of bacteria promote plant growth is production of IAA during the phosphate dissolving process in liquid Pickup-Skaya. The 3 isolates were detected to have the ability to produce IAA, the highest IAA. Production was produced isolate EF and AP5. Several factors influence the production of IAA by bacteria, which is a type of isolate. In this study, the media used did not contain tryptophan. However, the 3 types of isolate were able to produce IAA, even a small amount. The bacteria produce IAA without tryptophan using the independent tryptophan pathway, like Bacillus or Escherichia coli. The ability of this bacteria to produce IAA in the absence of tryptophan by using other aromatic amino acid as a substrate to produce IAA, such as phenylalanine, tyrosine, and even aspartate. The conclusion of this research is 3 isolate bacteria can dissolve phosphate solubilization in solid Pickup-Skaya medium in different varied activity. EF and AP7 isolate could dissolve phosphate quantitatively the highest phosphate solubilization until 392.87 milligrams per liters. And the maximum phosphate dissolution of this bacteria, 3 bacteria in the pH range of 4.19 until 6. Besides these 3 isolate can produce IAA without using tryptophan for the FNAP5, 7, and 10 respectively. So this is my presentation. Thank you for your attention. I would say thank you. Assalamualaikum warahmatullahi wabarakatuh.